Hello everyone, welcome to my channel. Today we are here with a new topic of pharmacology. Now we are going to study a very important topic about clinical trials. So now before studying this topic, we need to have some basic concept. If we see till today's date in market, there are thousands, millions of drug available in market. Now the question is how this drug came into the market. Like suppose in my laboratory, I am doing some research and discover one new drug. According to me, it's a very good anti-hypertensive drug. So what I did, I made 10,000 pills and took it to the chemist and said, please sell it. Can I do this? Obviously, the answer is no. Why I can't sell? Because it is a chemical compound. It can cause any harm. If somebody dies, then who is responsible? So obviously, before any drug come into the market and can be sold, there must be extensive testing on the drug. Basically, we have to test two things. First is the efficacy, and second is the safety. We need to know the drug that we have discovered is effective or not. Also, we need to know the drug that is safe or not. So, what we will do, we will check in animals. We will give this drug to the animals like a rat, rabbit, mice, guinea pig. So, we will test them to see how effective it is. Basically, efficacy is animal models. Model means that we first induce a disease to the animals. Suppose if we have to test an anti-hypertensive drug, so first what we will do, we will make a rat hypertensive. So there are lots of ways to make a hypertensive. If we perform any experiment or test in animals, it is called as a preclinical studies. But if we perform the test or experiment in a human beings, it is called as a clinical studies. As you know, in today's date, animal studies, how much they are regulated. So there are many regulations. So in preclinical studies, we are not supposed to take any animals and give drug to anyone by our wish. We need to follow our proper guidelines, and the guidelines are given via CPCSEA, Committee for Purpose of Control and Supervision of Experimental Animals. So this committee will give us a proper guideline, which animals we will take, how many animals we will take, what can be done, what cannot be done. So all these guidelines are given. So under CPCSEA guideline, we will do a preclinical studies. So let's assume we did an experiment in animals. It may be a rat, it may be a mice or a guinea pig, and we got a very good result. And we saw that the blood pressure has been reduced, and we confirm that drug is effective. And along with this, we need to see the safety as well. For safety, we will check the blood profile test, we'll check a kidney, we'll check a liver, we'll check a heart, and we found that all the organ is safe. So that confirm the drug is effective as well drug is safe. So what we did again, we made a 10,000 pills we have taken to the chemist and said, hey, now you sell it. Can I sell now? Obviously the answer is no. Why we can't sell? Because we have tested this drug in animals, but we have to give this drug to the human beings. So it is not guaranteed that the result shown in animals is going to be the same for a human being as well. So that means we have to study first on a human beings also, and it is called a clinical trial. So clinical trial is a testing of a drug in a human beings. So clinical trials are also have some guideline called as a good clinical practice guideline. GCP guideline. So in clinical trial, whatever we are doing or wanted to do is need to do under the GCP guideline. So one important questions can be framed here that the good clinical practice guidelines are not applicable to which studies. It is a animal studies. Because for animal studies we have a CPCSEA guideline. So for this what we will do, we have to study in a human beings. It is not like that you can take any human beings according to your wish. Before you go for clinical studies, you need to have some license. For this there is a licensing authority. By name itself you know that authority that has a power to give a license. So the licensing authority will decide to give a license or not. If you get license, you can move forward for a clinical trials. So depending upon the country, there are different licensing authority. For example, for United States, there is a FDA and it's a worldwide. But if we talk about India, the India's licensing authority is a DCGI, Drug Controller General of India. This work in a CDSCO, okay, Central Drug Standard Control Organizations. So for Nepal, we have a DDA, Department of Drug Administrations as a licensing authority. So depending upon the country where you want to do a clinical trial, you need to take a license. So we can say this licensing authority is the organizations that looks at the clinical trials. So for getting license, you need to put an application called as a INDA, investigational new drug application. So along with this application, you will submit all the preclinical studies report to the licensing authority. They will review this report. If you can convince to the licensing authority that your research might give a potential positive outcomes, they might give approval for the studies. If permission is granted, then we go for our studies in humans. And the clinical trial has a many phase, and the important phases are a phase 1, phase 2, phase 3, phase 4. So now we'll see the phase 1. So in phase 1 trial, what we will do, here we'll take a healthy human volunteers. So now question is, we are supposed to give a drug to the patient, but here we are recruiting a healthy human. So what is the logic behind testing a drugs in a healthy people? Do you think the healthy people won't have a side effect? Definitely, there might be a risk of a side effects. The basic logic behind taking a healthy people is that a healthy human have a good immune system. Suppose if any drug can cause side effect, the healthy human can tolerate easily. But if already people have a weak immune system, in case if drug can cause any reactions or side effect, the person might cannot tolerate it and might cause a serious problem. So that's the reason we do clinical trial phase 1 mostly in a healthy human being. But some drug has a very toxic effect, like an anti-cancer drug. We know that the anti-cancer drug mostly cause a bone marrow suppression. They might affect a healthy person as well. So this drug, which we already know they are very toxic, we avoid testing in a healthy people. So the next question is that what we mostly cannot do in phase 1 clinical trial. The answer is we cannot test the efficacy in a phase 1 trial. The simple reason is that in phase 1 trial, we are taking a healthy people. Suppose we have a new drug, for example, anti-tuberculosis drug, but we have taken a healthy people. If person does not have a TB, then how we will know that TB is cure or not? So it is very important to remember that phase 1 is not for efficacy. If it is not for efficacy, then for what? So obviously it is for a safety. So what we do in phase 1, basically we call a healthy man. We give a drug and we'll see whether drug is safe or not. So let me ask the next question. How many humans have received this drug before? Obviously the answer is 0, because the very first time we are going to give a drug to the humans. If we are giving the drug for the first time, how we will know how much dose should be given and who will tell? So this is what we determine in phase 1 trial. Basically, we have already conducted studies on animals, so we already know what dose was used in animals. So using that, we can apply a formulas to derive the human dose, which is called as a human equivalent dose, meaning that the dose that is equivalent for a human based on animal studies. However, this dose fell within a range, say 50 to 500 mg. Now the question is, should we give a 50 mg or 100 mg or 500 mg? Who will decide this? So this is determined in a phase 1 trial. Essentially, we invite a healthy volunteers and give them to the lowest dose in the spectrum. Let's take example 50 mg and then conduct a extensive testing. We'll test a hematology, we'll check a RBC, we'll check a WBC, we'll check a kidney function test, liver function test, ultrasound, CT scan, everything we'll do. So if no issues arise, we send that person at a home. So next again we invite another healthy volunteer and give a higher dose, example 100 mg, and repeat all the tests. If there are no problems, we move to the next volunteer, increasing the dose further. So this process continues until an intolerable side effect appears. So this process continues until an intolerable side effect appears, which means the dose cannot be increased beyond this point, and this is called as a maximum tolerable dose, MTD. So the main aim of phase 1 trial is to determine the maximum tolerable dose, the highest dose that can be safely given. So beyond this dose, no further trials will use a higher dose. After phase 1 trial, we determine the maximum tolerable dose. Let's take example 500 mg. This means that in all future trials, no dose will exceed a 500 mg. It will always be lower. Okay, so the phase 1 trial helps us to determine the maximum tolerable dose, ensuring the future trials remain within the safe limit. So in next class, we will see the phase 2, phase 3, phase 4. Also, we'll talk about our zero phase. Also, we'll see our post-marketing surveillance. Thank you. Thank you for watching.